Okay, I think we can probably go ahead and just get started for time's sake. And hopefully Baboo will be able to join us shortly, and we'll be able to get to that. But I think we have quite a bit of interest here about what's going on with people. So thanks everyone for attending today's Beyond the Scope. I'm kind of excited about having a different panel discussion, discussing kind of why CMAS and all that kind of stuff. So today we have Charles Bell. He's an associate professor in the College of Medicine. Baboo, he's a research professor on material science engineering, and he has an office here at CEMAS. So he's around quite a bit and usually on an instrument here. And it's fun that we get Austin here as well. Austin's basically at CMAS most of the time as the application specialist from Thermo Fisher Scientific working on the TEMs. We'll have a couple upcoming talks as well. So in 2 weeks, we'll be talking about EDS in the TEM with Hank, and then we'll be back to talking a little more about the HeliScan with Carly. After the schedule, we're probably gonna be adjusting our schedule a little bit to be more of a monthly schedule as we go forward with the discussion series. So a few things that we're going to do. We just want to talk about basically with these people, what brought them to CMAS and kind of how CMAS is unique. And we want this to be very interactive with the audience. So if you have any kinds of questions, feel free to just throw those in the Q&A right away, and we'll get to those after we've had a couple other questions, or you can also put some information in the chat as needed. We can also, if you want to talk to them directly, we can basically allow you to talk, and that works pretty well. So I just want to mention a couple of the resources that are available at CMAS if you haven't been to CMAS before. So we've been doing these Beyond the Scope talks for about a year now, and we have quite a library of them online. So if you go to CMAS, you can go to the YouTube page and view all of the old ones. If you want to talk about any of the resources we have, feel free to talk to any of the instrument managers. All of our contact information is on a CMAS webpage. Or also, if you're interested in doing any remote work, we do quite a bit of that. You can email CMAS as well to get in contact with the right person doing analysis or other work. And again, if you haven't been to the facility before, we have a really cool facility here that's been custom built to really do a nice job working with all these instruments. So with these instruments, we have things ranging from a couple aberration corrected Titan instruments and some other TEMs as well for other analytical applications. One of our newest growing things is going to be the cryo-EM side of things, which we have our Glacios and Titan Krios for any of that. Moving on to the other side of the building is where we have all of our small beam instruments. So either our dual beam instruments with our Helios and Nova, or all of our scanning electron microscopes, which is what I'm heavily involved in. And besides the electron microscopy, we do X-ray imaging with our HeliScan, a lot of XRD analysis, and there's a bunch of other capabilities, including nanonotation, available as well. So to start the panel off, I want to have all our panelists introduce themselves a little bit. So we'll just go across the board right here. We'll start with Chuck with College of Medicine. So if you want to go around, introduce yourself, kind of talk about your position a little bit, what you use at CMAS. And one thing I always like about what we've done with Beyond the Scope is say something that you like to do outside of the lab.
Okay, yeah. So my name is Chuck Bell. I'm been at OSU for 20 years in the College of Medicine, Department of Biological Chemistry and Pharmacology. I'm a structural biologist, so we try to visualize macromolecules, proteins, protein DNA complexes, and solve their atomic structures in 3 dimensions. And the way I was always trained to do that was with X-ray crystallography, and we still do that. There's a facility on campus in Rife building for macromolecular X-ray diffraction, and that's still a great technique. But in recent years, there's been a boom, obviously, in the cryo-EM, where you can now get high resolution, very similar to that of crystallography, but without having to grow crystals, which can be a bottleneck. And so CMOS got the equipment for this 2 to 3 years ago, I want to say, and thankfully we've had CMAS on campus to house all that equipment and have the infrastructure and the personnel and the support expertise to to get those instruments up and running and maintained. And I don't know what we'd do if we didn't have CMAS had already been here and established. So, but I've been using the Glacios and the Titan Krios at CMAS for the last 2 years. We've solved a structure at about 3.2 angstroms resolution, so we're getting ready to publish it, and we're real excited about the data. It didn't happen overnight, but it can. If once we found the right sample, essentially, it worked very, very quickly. So yeah, I've been very happy and excited, and with the cryo-EM facilities at CMOS. So, oh, and outside of the lab, I play tennis, and I got a new puppy who's might hear him in the background. I thought he was going to be well behaved, but we'll see. Okay.
Awesome, thanks, Chuck. So next up, we have Baboo. I saw he was able to join now. So Baboo, are you there? If you can give a quick intro to yourself and talk a little bit about kind of your current position and some of the resources you've used at CMAS. And then I always like to hear about what you do outside the lab as well.
All right. Hey, thanks, Dan. It's wonderful to meet you all. It's a little smaller gathering, and most of them are from CMAS. I just see that. Let's see. That's a good thing. So, so let me start with what I do outside of the lab. In fact, I do everything outside of the lab now because you can't go to the lab these days. So my position here is a research professor. It's a full professor position, except that I had to bring in my own money for most of my efforts, including my salary. So that's been, as you know, if you're a research professor, you know how difficult it is. But fortunately, managed to do it so far and support a small group of people who actually work with me. I'm a metallurgist by training. I mean, I've never deviated away from metals all my life since my undergrad days to now. So I've been at OSU for past. I was in OSU before as a postdoc here. I graduated from University of Cincinnati with a PhD in material science. And I went to work for the Air Force for a while. But then I came to Ohio State as a postdoc. Then I went to Air Force for a while. And then the CMAS came, which is the biggest attraction to come back because I'm a microscopist by training. But I use microscope to probe the metals. So I'm a physical metallurgist by profession. So I use, and without CMAS, I wouldn't be here to attract so much funding from outside. So this is the biggest attraction for companies to interact with. So I work on high temperature materials. I work with, when I say high temperature materials, is aerospace kind of materials, specifically aero engine materials. So I'm among the few who had simultaneous funding from all three major aero engine companies. I had funding from Rolls-Royce, Pratt & Whitney, and GE at the same time. So seems like they trust me here, right? And I've been able to do, I mean, when you work with the companies as opposed to work with the government funding, which I also have done before, the companies are very focused on what they want to really get out of the funding. So it's much more difficult to work with the company because they have target dates and everything. And what they do at the macroscopic way, you know, over the years, even more is they've come to understand that what we see in very small dimensions is makes huge difference for them for the macroscopic properties. So, for example, if you look at the last month, You might have seen it on TV that there's an aircraft, right? I think it's United Airlines, I think, because the engine got fired when it took off from the Denver airport, right? And it was all over the TV, right? And those are the tragedies can happen if some fundamental properties are not understood. Because in that incident, you can see the whole fire, the engine fire, and that because one of the fan blades, like you see from the outside, it took off and just came off and then went through the engine, and that created the fire. And that happens to be a Pratt and Whitney engine. So we have some collaborations, and OSC is a big collaborator with the Pratt and Whitney. If anybody wants to, they have what they call a RUP center at Ohio State. It's at the Ohio State Airport facility. They have underground facility where they do the live testing. Some of those, they have the wind tunnel, they have high temperature erosion. So it's a huge facility there. So we are a small part of it because they come to CEMAS, and everybody in CEMAS actually help us with the characterization. So going back to what I said at the Denver airport, it is a titanium blade, we think, because there's not, the NTSB has not declared what the project is, but we think it is the titanium blade came off. And that is because what we call the titanium blade is outside. It's not even a high temperature. So what we call cold fatigue, we call cold 12 fatigue. So that's an effect that's very intently looked at by the aero engine companies. This is a titanium who actually creeps at room temperature. That means it increases its length in all dimensions actually as a function of stress and temperature. And you know how small the clearance is in the engine, a couple of millimeters, even less than a millimeter in some part of the engine. So we cannot afford to have any part extending from beyond a couple of millimeters. So I think that's what is probably we are thinking is happened to this machine, so the engine. CEMAS is a wonderful place for them to come and collaborate with us. Like we can tell them at the smaller dimensions what is happening, and then they can relate this to the property. So it's a wonderful experience, and then for me to be part of the CEMAS effect and part of the MSc. I'm part of MSc here. So it's a wonderful set of faculty. If I had, and all the CEMAS staff, they all hold doctoral degrees. So they're all well educated and very expert in their own field. So it's a great place to be bounds of ideas, and then we work as a collaboration. And if you ever visited CEMAS, you know how close we are to each other. And then the one thing I would like to stress is that CEMAS has a room as all the graduate students sit. Our students sit there. Now these days, last 1 year, they're not able to do it. Prior to that, that is where the ideas come from. They talk to each other, they help each other, which I've never seen before the CEMAS. Each group has got its own little place, and then they interact among themselves. But CEMAS has provided this opportunity for the graduates. It's about 75 desks there. They all hang around there, and they learn from each other. That's a wonderful thing what CEMAS has provided. I'm so happy to I'm here. Sorry, it took more time than necessary, I think. Thank you.
Thanks, Baboo. I think you hit a couple of questions already. I was going to think about asking, so I took a little bit of my thunder there, but we'll get around to all that as well. But no, I really appreciate you hitting a lot of different topics there. And I think that could start some other discussions as well. If people have questions on any of that kind of stuff, feel free to start putting those in, and we can get to those as well. So
before I finish, one last thing. I'm kind of disappointed that nobody is wearing the Ohio State red t-shirts today. Maybe Austin does a little bit. So it's a thing in my group that on the Friday we have to wear the red shirt. Otherwise, there'll be penalties in the evening beer session we have.
Nice. Okay, so next up, Austin, you want to talk a little bit about yourself?
Sure. I'm Austin Wade. I'm an application scientist with Thermo Fisher Scientific. I'm based out of the Americas Nanoport in Hillsboro. That's where the remainder of my group resides. But for about 50% of the time, I'm in and around CEMAS. This last year, obviously, I've been here significantly more. So I've been here to provide any sort of application support or help with any number of the TEMs that we have here. And the TEM is my specialty. Previously to working for the Americas Nanoport, I was still employed by Thermo Fisher Scientific, but I was at the University of Manchester in England. There working in conjunction with our R&D department in Europe for a number of years. And then when the opportunity came to come back to the States, I jumped on it and got a position here at in Ohio. So basically, I'm here at CEMAS to help with any and all TEM related questions, issues, experimental concerns, any number of things. I'm particularly excited about forming any kind of analytical spectroscopy with the aberration corrected systems here. So that's where my primary interest resides. And outside of research, I'm an avid fisherman, so I try to do as much fishing as I can. Being in Ohio, I don't get out on the ocean as much as I would like, but I transitioned to more freshwater fishing. So that's what I do for fun.
Awesome. Thank you, Austin. So kind of where I want to start this, I think Charles and Baboo hit on this a little bit, but kind of talking about impactful projects that you worked on here at CEMAS, and also kind of what instruments have been able to really enable that research here. And I think we'll start with Charles again, if you want to talk a little bit more about that, and Baboo, then Austin again.
Okay, so you mean the instruments or the science behind the project? I'm sorry, which?
Yeah, so you can talk a little bit about the science behind the project, because
Okay.
that's what brings everyone here, right? You have your science and you have a problem, so you're trying to figure out how to solve that problem. So it's nice to have a little background on it, and then what kind of problems were there that we were able to help with, or even not help with.
Yes, so we study, protein DNA interactions and that are involved in DNA recombination, and they are used for DNA repair, so they can be involved in cancer, either causing cancer or treating cancer, but as well as genome engineering. So in bacteria, you can electroporate DNA inside cells, and our proteins that we're working on will bind that DNA and incorporate it into the genome to make changes. So I'm sure everybody's heard of CRISPR-Cas9, probably. It's kind of similar to that, but it only works in bacteria, but it's super powerful and exciting. And the protein itself it has a molecular weight of 30,000 Daltons, so 30 kD, what we say. And it's kind of a messy in terms of trying to get crystallization. So the protein will coat DNA, and it's not the type of thing that would crystallize easily. So if we have like an 83-mer nucleotides of DNA, each monomer of the protein will bind to 5 nucleotides. So you'd have about 16 or 17 monomers bound along a 83-mer DNA. And this just simply wouldn't crystallize. And with the cryo-electron microscopy, we prepare that protein DNA complex and freeze it on a grid with help from Giovanna, and use the Vitrobot to plunge freeze it very rapidly into liquid ethane, and then screen that grid on the Glacios instrument, the TEM, while it's frozen. And we tried a lot of different samples, but one showed these remarkable helical filaments, and we were able to then collect high resolution data on the Krios, always with help from Yoshi in the Glacios and the Krios. And my student was doing all this, Brian Caldwell. And I should mention, actually, the CEMAS has a wonderful course. I forget the course number, but it's Materials Science and Engineering for all the graduate students to take, to really learn the ins and outs of how a TEM works. And Brian took that course and really got outstanding training on how to use the instruments. And he's definitely better than I am now at using the instruments. And, then for cryo-EM for proteins, there's a lot of software involved, and CMOS has a computer with the software that's available for use, but my own lab now has 2 computers. And so after we collect, the data collection happens in a day, but there's a lot of troubleshooting and sample prep that might happen for a couple weeks. In our case, it took a couple years to get the project, mostly on the sample end, just right. But then the computer part happens. It's called single particle analysis. You need GPU-based computers to get it to work. And eventually we got a very high resolution structure, and it's just super exciting when you see the 3-dimensional reconstruction, or we call it a map, and build an atomic model. And the resolution is about 3.2 angstroms, so we can see all the different side chains and atoms of the protein molecule. And it's something that you could not have done with traditional method X-ray crystallography, but we were able to do with the high-end cryo-EM instrumentation at CEMAS. So anything more I can tell you about that, or is that kind of what you had in mind? Or
Yeah, no, awesome, thanks.
Okay.
It's my side of things is always going to be on more material, so I like always hearing about the bio side, what's been going on there and how it's been going, because it's very new at CMAS, so it's great that it's taken off so well. You've put in a lot of work there as well.
Can I ask a question to Professor Bell?
Sure.
Yeah, definitely.
Yeah, no, I'm in the physical sciences, and then before CMAS, when the cryos and glaciers came, I never interacted with the bioscience people. It's wonderful to hear the other side. And I read about a CRISPR. Can you say something more about it? It's a gene editing.
Right. So CRISPR-Cas9 is basically it makes double-stranded DNA breaks in cells, and at very targeted locations. So it's RNA guided to make a break in DNA. So it's a protein, but it binds to RNA. I'm not sure if everybody's familiar with RNA versus DNA, but DNA, of course, is your chromosomes that encode the genes, and then there's an RNA copy of the DNA is made. That is usually the messenger that can then make the protein. But anyway, CRISPR is everybody's sort of, even my elementary school age kids have done research projects on CRISPR, I think, and it allows you to make breaks in the DNA, which can knock out genes. So or you can use it to make variants of genes. And it works really well in human cells, and it can be introduced by viruses. And so it has really revolutionized the way people do genome engineering for human cells. But my protein is actually more powerful, I would say, than CRISPR, but it only works in bacteria. So you've heard less about it. The word you'd hear is recombinering, or red recombinering. In bacteria, but people are using it to do interesting things with bacteria, such as remediate toxic waste or make molecules. People are trying to engineer bacteria to hunt down and kill cancer cells. So all kinds of different things that people are engineering bacteria to try to do. Our protein is very much involved in the manipulations of the genome there. Yes.
That's awesome. Yeah, the first time I heard about the CRISPR was on the CBS 60 Minutes. They had a program on sickle cell anemia. They solved that thing using CRISPR. So that's one. It's awesome. That's what Dan. This is awesome. Like, I would have never been able to talk to Professor Bell about this without this kind of a forum.
Yeah, no, I think it's fun having a forum here. There's always that interaction. I think CMAS started off kind of before, started off in material science and engineering, right? And now it's the whole College of Engineering, and bringing in bio side of things, it really brings the material scientists and biology together a little bit. So we can hopefully be able to have some more interactions soon in person as well. So that'll be good.
Yes,
so I'm gonna change a little bit. So Austin, kind of a similar thing. You're not as involved with science and people here, but I know I've heard a little bit that you've been able to get involved working with users as well here. So if you want to talk a little bit
Sure.
about either working with the users or also just kind of working with the newer instrumentation here as well.
Yeah, one thing I can mention was a pretty successful collaboration or fulfillment of my role here at CEMAS. There was a project that's actually still ongoing. We're looking at imaging really small scale magnetic domains in materials. And one of the traditional ways of doing this is to use Lorentz microscopy, where if you don't know much about an electron microscope, all of the lenses are electromagnetic lenses. So as you can imagine, there's a lot of different fields involved in the microscope, pretty strong fields up to maybe about 2 Tesla. This is not good if you have a magnetic material. If you have a soft magnet and you put it through a field, you can change the structure. And if you're looking to investigate those structures, that's obviously not something you want to do prior to imaging with the microscope. So traditionally what you do is you use something called Lorentz microscopy, where the objective lens, or the field that the sample is usually sitting in inside the microscope, is greatly reduced to 0. So you have a field-free environment for the sample. And then below that, there is a Lorentz lens, which becomes your image-forming lens. Traditionally, the objective lens is the image-forming lens, but when you turn it off, you have to use another lens. Now, to look at these materials, the information that they were attempting to gather was essentially a phase reconstruction, or something like a phase reconstruction with integrated differential phase contrast, which is only available in STEM. So not conventional TEM, but a STEM mode where you use a small electron probe that you scan over the sample. Now this has the effect of essentially optically turning the microscope upside down. So a traditional Lorentz approach is not applicable. But what we could do is change the, still run the microscope in Lorentz mode, but use the mini condenser lens above the objective in the same manner that we would have used the Lorentz lens in traditional Lorentz microscopy. The microscope is not designed for this. So my role here was facilitating the microscopes at CEMAS to be run in this mode. And this was a real, a good effort in collaboration between the users and the staff at CEMAS, identifying a need. And I helped implement that need with support from our research and development team in Europe to have a successful Lorentz STEM mode at CEMAS that we're hoping to use in conjunction moving forward to combine this for STEM investigations of these small scale magnetic or electrical domains and materials, and combine that with the dedicated 4D STEM detector that we have here at CEMAS to really see if we can get the most out of this type of investigation, this type of microscopy being performed in STEM on these materials. So it's really quite exciting, and it was nice to see everything come together in a way that you know, everybody benefited, and it wouldn't have been possible without the facility such as CEMAS, and my presence here was great to help that come through.
Well, thanks, Austin. Yeah, no, I think that's one thing I kind of appreciate a lot about CEMAS coming here is we have a great relationship with Thermo. If anyone's been here, you'll notice all of our equipment is from Thermo, and we have really good interactions with them all the time. I'm emailing them constantly to try and push the science as well. So having Austin here on TM side's been really great on that front. And then, yeah, Baboo. So yeah, you talked a little bit about some collaborations and projects that you're working on at the start. What interesting things have you seen in there? What would be one of the top projects that you probably worked on with those that you could talk about? You're muted, Baboo.
Sorry. So I can talk about a few things, not everything, which with the project that we work with the aero engine companies. But for example, the high temperature section of the engine, what we call nickel-based superalloys, that can stand high temperature and high stress. And this has been the development in the industry over almost say 60, 70 years. So, but they always make constant improvement for newer alloy design. Uh, so, uh, so, so traditionally we say the paradigm was that, okay, the defects during the formation or during the service is bad. Um, but sometimes the defects that we cause during the last 5 years or so at a higher state, especially that some of the experiments done at CEMAS has been really helpful to change the kind of, for example, if you some of these temperatures, some of these materials at intermediate temperatures. When I say intermediate temperatures above 700 degrees centigrade, they deform and they introduce defects, and as a consequence, what we call stacking faults. So these defects are there in the material. As the material is deforming slowly, they develop, and depending on the alloy, these defects could be strengthened because of migration of other what we call alloying elements or species to the defect, and they kind of stabilize the defect, and then they restrict further deformation because of the diffusion of specific elements to the defects. So as you would normally say, okay, because the defect is going to fail, it's going to deform faster, creep faster. But in this case, actually, not all alloying elements, but certain alloying elements can go and then stabilize these defects, and then not propagate further. So this would have been never possible without the atomic resolution, the instrument that we have in CEMAS and elsewhere. And so this was actually done in CEMAS, and Professor Mills' group started this as part of this in the early time before I went to the Air Force Base. And now it has actually developed into a big program with the Indian company. They're looking at changing the alloying elements, but everything depends on what we see in the microscope. And then and then when you know the microscope, when you look at the atomic resolution microscope, you know the atomic structure, okay, but you don't know what the species of the elements there, what kind of alloying elements are there at the defect. The defects are actually 2 or 3 atomic layer thick. So imagine how complicated it is. And you're not looking at one atom, you're looking at a column of atoms. So we do the what HADF STEM, the high annular diffraction. And there are so many detectors in THEMIS and the Titan there that we could use image differently. And then, so based on that, we know what elements are segregating to that. And then based on that, in the locally, what would be the local crystal structure? Now it is atomic, things change, so it could be a little more ordered structure. So that area is developing, and some are bad, some are really good. So based on that, this actually directly goes back to the alloy designers, how to change their alloy compositions. So we've been very successful in actually, at least telling them what works and what not. And then so those alloy designers started taking the note of it. So when we do it at this level, we have to be useful to the industry, so they can think about it more, because those are the ones actually going to use the facility. I would say, if I can, one more example is that usually these materials fail at extreme temperatures. Is that we start with a small crack. And then when we say a crack, actually, you see it in the optical microscope, it'll be like, 0.5 millimeter or less than 0.5 millimeter. But in atomic dimensions, it is an atomic dimensions in the crack nucleus. And then, so what happens ahead of the crack? So those are the things we're carefully looking at, what we call focused ion beam. So we actually able to prepare a TEM sample right ahead of the crack, just a few nanometers ahead of the crack, and look at the atomic dimensions and look what happens to the morphology. Changes to the chemical nature of the region ahead of that, and also the structural changes ahead of that. When I say structural changes, in terms of defects, what can cause. So that has been always the problem with these instruments. So this has really improved our kind of capability to probe very small dimension and where we want to see the changes happening.
Nice, thanks, Baboo. So you mentioned quite a few instruments. Just Do you have a favorite instrument here at CMAS? I know you've been on basically all the different microscopes. It could either be an old instrument that we used to have here or a current instrument.
Yeah, right now my favorite instrument is CEMAS. That's where I want to be. This has got all the bells and whistles that I need to and uh, and and of course the other thing actually which is I wanted to say about what the first time we had the in situ capability in the SCM level is what actually you have, Dan, the Quattro, which you and I are planning to do some experiments.
Mhm.
So that is a very special instrument which many places don't have it, and it'd be interesting to see actually if you, what it means by that is that you can you can load a sample and look at it in the SCM as you deform the sample or whatever rate you want to deform the sample, and you can look at the high resolution what is happening when the defects emerge from those. And also it has got it got a certain temperature capability. Basically, you can kind of heat the sample up, and which is really necessary. And I also can get optical, like Dan is good at that. The optical signals that you get also you can give you the other one. So I think eventually I think the Quattro is what we want to use it for. One of the projects that I have from Exxon Mobil, which is pipeline steel. So they have interested in looking at certain things as a deform. So I will be using the Quattro in the next few months or so once we get the samples ready. So my my favorite instruments right now is everything except glaciers and cryos because I don't work on those machines.
Yeah, thanks.
I'm not a and I'm not a biologist, so but I'd like to learn more about it because they have so many interesting things going on there.
Yeah, I've always thought like the cryo instruments, they're dedicated
Yeah.
for bio, but I think there's probably other applications that could be useful. But no, I think it's cool you hit kind of on the topic of your favorite interest being the themis, where you can do that really high resolution work. I love seeing that kind of stuff here at CMAS. But then also just kind of how you backed it out to my favorite instrument, obviously, is the Quattro, where I manage and work on quite a bit. But where we kind of can do that whole kind of length scale from looking at that macro scale, deforming, and then going in through that. Like I've seen a few projects that are nice in there, where you go into the fib, lift it out, those defects, and move them in, and go on to your favorite instrument on the themis now. So no, I'm glad you mentioned that.
Yes.
And so, Charles, I want to ask you, you talked a little bit about how much you've worked with Yoshi and Giovanna on different projects. So I don't know if you want to comment a little bit more on how useful you found the staff, if there's, I was like deficiencies as well and things, but just kind of benefits of the staff that we have here at CMAS, how that's helped you and could possibly help other people as well. So I think that's one of our big strengths as well, besides having a ton of instruments, is also a lot of very capable staff here.
Yes, absolutely. I mean, I think the first person, if I remember correctly, was Hank, and I've been amazingly impressed with Hank's knowledge, and I got to get him to talk down to my level sometimes. But he's just so impressive with his knowledge of the TM and how it all works. And I mean, the course that I believe Hank and Sarah and Yoshi have taught is just really valuable resource for the students. So, but yeah, and then I remember I think when CMAS got the equipment, and then we're able to get Yoshi to help you know, keep uh, keep track of the instruments and train the users. And so she's been you know, very helpful and patient, and you know, I don't know what we'd do without her. You know, it for me, I think my student has finally got it down where he can practically go in, and at least on the Glacios, independently, and for the most part uh, operate the instrument. But there's a lot to sort of keep track of, and so you know, make sure that everything's aligned fully and that you're doing all the right steps, especially with the Krios, that it's more involved. And so, we've been very happy with our interactions, and it's kind of a the users need to get more independent and be not need their hands held so much throughout, but really it's the type of thing that for me do it every couple months or so, so it's really hard to learn. It's not quite like driving a car or riding a bike. It's more um, we need help, and that's CMOS really has great staff. And then Giovanna um, was really had a lot of expertise in the sample prep and applying sample to the grids and freezing them. It's really half art, half science, and really takes a lot of experience and troubleshooting. And so she's been, worked with my student Brian a few times, and I think Brian, now I need Brian to choose to train me all he's learned from Giovanna, all the tricks involved, and getting a really good clean grid is quite it just takes a lot of experience and a lot of expertise. And so she's been great on that end. I think she also now helps um, Brian get images on the glacios. So everything's been great and really phenomenal expertise. Again, I don't know what we would have done on campus if somebody would have given us $10 million to get, all the cryo-M instrument and just said, here, go set it up. It would have been impossible. So,
Perfect. Thanks. Yeah, no, again, yeah, I think it's really cool seeing that build and just the interactions that I have with them. I've learned a ton of stuff from them and all that. Yeah, like I said, sample prep, just that experience there is crazy getting that kind of stuff done. So I know, sorry, 11:45. But yeah, if anyone else has other questions, please feel free to start putting those in the Q&A or chat. I still have a few questions for everyone else, but if other people are interested in other things as well, feel free to chime in a bit. But I kind of want to ask Austin a little bit about the facility here. So previously, before I was here and everything, CMAS was a different facility name, and then it came to over here on West Campus and fully designed facility with Dan Huber, everyone put in a lot of work to figure that out, how to make it work. So I was wondering if Austin could talk a little bit about what his thoughts are on like the facility design. Can compare it a little bit to other places as well if you want to.
Sure. One of the best things from my standpoint being with the company that sells the equipment that it's used here is that CMAS is a purpose-built facility. So it was designed for electron microscopes for their installation, for their upkeep and maintenance. And it really makes a lot of our job keeping these microscopes running, troubleshooting any issues, everything so much easier to help keep improve the uptime of the microscope. If you compare it to other facilities that aren't purpose-built for EM, you'll notice really if you go to any other location that doesn't have its own standalone microscopy facility, the microscopes are always in the basement. They're usually in less of a accessible location, and you might have to live with certain inconveniences like other things that happen to be in the basement. There's often machinery, there's often loading docks, things of that nature, air handlers, all these things detract from the environment that the microscope works best in. This is usually done because you have some of the best locations in basements of buildings. They're stable. It's easy to get this equipment in and out, or easier than if it is on a higher floor. And you can put the infrastructure around it that's needed in terms of water lines, air lines, anything that you need for the electrical connections. It's always easier. But in a purpose-built facility like CMAS, the rooms were designed with the foreknowledge of what was going in them. So we have anti-vibration pads that the microscopes sit on, so you don't have vibrations coming through the floor or from other rooms into the microscope and then distorting your image. There's dedicated water line. There's a room down the hall that has all of the chillers lined up in a row. So one, you can access the water, you can clean it, you can do whatever you need there. And two, you don't have all of these heat sources in close proximity to your microscope. So this is all done with an eye towards keeping everything stable and making maintenance of the microscope very easy. Any service actions are done quickly, and everything's really accessible. And for the user, now you might not notice it going between instruments here at CEMAS, but if you took data that you collect here at CEMAS and compared it to data, same instrument on a different system, you'll probably see cleaner data here. And that's largely due to the stability that's achievable on these microscopes, because we have everything taken into account when the facility was built. So I hope that answers your question.
Yeah, no, I think that's great that you're able to talk about a lot of the things that have been developed here. I know when I came here to visit, when I was going to start a couple of years ago, I was really impressed with the facility, and that's one of the reasons why I definitely want to work here as well. And so, Baboo, I know you talked a little bit about it as well, of some of the things that have changed at CEMAS instrumentation wise. So Themis, the Quattro, all that kind of stuff that's come in the last few years. So what would you say are like the things that make CEMAS something that could be a leader in the field of microscopy, and what could also be improved as well? Like anything that you would be really, really interested to see here at CEMAS?
Oh yeah, I think Austin kind of touched a little bit on that. That see when you go to other places that you see the instrument in the basement, maybe it belongs to the one department, or so. But having a separate facility like this, it really helps. Kind of not only doing the experiments, collaboration, everything really helps. So that way, I think CEMAS is very unique in the country, and very few places actually have this kind of and also especially to have a collaboration with the leader in the industry, with Thermo Fisher, it really helps because they give us special attention. Like Austin being here in the facilities, we are so lucky to have him here. Many other places don't have. They have to still log in a request to have somebody come and look at it. So Austin has been I hope he wants to stay here for forever. So like that. In terms of improvement, and I think it's a constant. Maybe we can always, and the one thing we would prefer as a users is that we want the facilities to be updated, the instrument, because even the Themis is new, right? But if you look at some of the instruments that there are additional instruments. I mean, there are updates by the manufacturers, right? There are something we cannot do here, but people who bought the microscope in the last few years, last year or so, have a little more capability. That is an ongoing thing. A few years down the line, we will upgrade it to new instruments, and we will be at the top. So I think our director knows those things in and out. So how to stay ahead in the game. You have to be having you have to have the top-notch facilities all the time, which is very, very difficult to do. You have to write proposals to get the funding, but that will keep us as the leader in like we are now in certain fields. So that'd be nice. I can't think of anything at this point for improvement, because I'm happy. That's good. And for example, even like in the last week, there was director sent out an email saying that we have this micro city. We want more users to that. And then do we want it? What do you want to do with the instrument? That kind of an input from the director asking directly the users is really helpful, saying that, okay, that means they gather support for what is needed and what is that to be there. So, and that I've never seen this in other places, that the users' input are so important for running the facilities. It's good. But I want the students to come back and then
Yeah, so I think that's great talking about. Like, there's always updates. We're constantly updating equipment, which I think is awesome as well. So, I think just for anyone that wants to talk about, we have about 5 minutes left. I didn't take you up all the time with some questions I can't ahead of time. But if you guys want to discuss a little bit about any new trends you've seen in microscopy, so it's maybe not stuff we have here, stuff we could get, trends where things are going, either material science, biological related, or even just R&D into instrumentation itself. You guys want, okay, any of you guys can start talking about that as well.
Well, I think from the bio end, looking inside cells, real frozen cells, cryo-EM tomography, where you take a single cell and rotate and collect images to get a three-dimensional image, that's going to be huge. I'm not sure all cell biologists on campus, neurologists that look at weird structures inside neurons and so forth, if they're really aware of the revolution that's going on with imaging inside cells at 8 to 10 angstrom resolution, for example. So I think that's a huge one, is the cryo-EM tomography to look inside cells. So on the bio end, yep.
Yeah, it's kind of amazing how the biomicroscopy works. And so when I go to when I go to M&M, I kind of make sure that I attend some of the keynote lectures in biosciences. And so when I did it for the first time many years ago, when the tomography was popular at the time for bacterium tomography and all this, I sat to the lecture. And when they showed the first few images, that okay, this is how it is, nice colorful images, for a physical science like me, I couldn't. I said, okay, what am I seeing? Right? It's such a blurred image, and then when they do the tomography, they develop into this beautiful bacterium. They had this complete. And based on that, it had this little two antennas trying to attach itself to the cell. And then they made they conceptualized a micro machine of bacteria micro machine, and it's absolutely mind-boggling how the tomography effects have taken into the sciences. I think so. I learned a lot quite from the biosciences. So I'm sure Professor Bell can. He knows all about this. But for us, it's really interesting. So. And when I teach microscopy courses sometime, and I see some bio students sitting in my class, and then they have no idea about the diffraction, about that, and they're really curious about what is diffraction, what is diffracting, where is it going, right? So they are also learning quite a bit from this. So at some point in time, I think it will be nice to collaborate with the. I think we're almost out of time now, but I want to ask you, Professor Bell, that we use quite a bit of energy loss spectroscopy to see energy loss happening with interaction with that. Is there anything in the biosciences they do a lot of energy loss spectroscopy at all? Any use?
I don't know. Yoshi might be able to answer that better than I can. I might know that by a different name.
Okay, right.
But yeah, I've not heard of that term so much, but it could be something by a different name that we do.
Yeah, it is definitely. It is. So when the electrons go through the material, it sees the individual atoms, and then by their interactions, whether it is close to the nucleus or away from the nucleus, then it loses its energy. So it could be coherent and incoherent when it comes out of the sample, below the sample. So there's a spectrometer that actually can separate out depending on how much the electrons lost their energy, and you can image them depending on where is it coming, what point is coming from, and also it gives you an idea about what species is present in. So I was just curious about, is there any use other than identifying what species is there in the sample? in the bioscience. I was just curious about it.
It's used to a degree for ZLP filtering. So the zero loss peak is selected for contrast enhancement in cryo DEM.
Okay.
You're always struggling for contrast with biological materials, so that's one way to increase the contrast and improve your throughput.
Okay, that makes sense. Yes, yes.
The only other example I can think of is I think like certain molecules that have like iron sulfur clusters or other metal centers. Sometimes I think people have tried to use some eels there, but it's very uncommon, I would say.
Okay. Yeah, our center director, Professor McComb, is an expert on energy loss spectroscopy. So, I mean, so when he came here, the whole game changed. We never did a lot of energy loss spectroscopy before he came, so he really made a big difference. So,
it sounds a little similar to, in crystallography, anomalous dispersion and diffraction from metal centers that absorb the incident X-rays. So we use that in crystallography to solve the phase problem for macromolecules. But yeah, I'd have to study that a little bit more. But yeah.
Okay, so it's noon. I think it went pretty quick, everything, but I won't be able to let you guys go. But do you guys have anything else you want to add real quick that we haven't been able to get to? We'll just go through. Charles, have anything else?
No, thanks for the opportunity to have this discussion, and it is pretty neat to have interact with the material scientists, and I think we can learn a lot from each other from our different worlds and trying to explain our science to a broader audience. And I think if we could get more of these interactions, in some types of forums where we're really engaged and go deeply into each other's science, we can learn a lot from each other, I think. Yeah.
Definitely. Baboo, you have anything else you want to add?
I think what you said is perfect. I ditto echo his sentiments. Exactly same thing. The more forum that we have, the more we learn from each other.
Yeah, no, that's one thing I've always enjoyed too.
And,
definitely.
And Dan, thanks for doing it, Dan. Really nice of you.
And yeah, Austin, you have any last?
Yeah, I think the collaboration is a good way to end. It's great that we have so many different disciplines involved with CEMAS. And if you're ever in the hallway and you have any questions, or there's anything we can collaborate on, help each other, feel free to stop and ask.
Okay. Yeah, thanks a lot, guys, for being able to take time for this. I really appreciate it. And I think we learned a lot and got to hear some interesting views on this. So thanks again. And I think, one more slide. So I just want to mention again, just for everyone still there, that we still have a couple more talks this year going on. So to finish up the semester, we'll talk a little more about EDS with Hank, and then Carly will be talking a little more about the Healy scan. And you can always go to CEMAS events to find those, or the CEMAS page, and there's a whole page dedicated to Beyond the Scope if you want to see anything else. So thanks again, guys, and we'll talk again soon.